So the topic is drug development. Drug development. So first drug is discovered, then in preclinical trials, animal testing is done, then human testing is done, and then it is published to market. In India it is CDSCO, and in US it is FDA. Drug discovery. So we found drugs from nature. In nature, in plants. So the Papaver somniferum, we gives morphine. Atropine is discovered in plants. Atropa belladonna. In animals, we can do vaccine. Heparin, tetanus toxoid. In microbes, we can found drugs like penicillin G and the height throughout screening. It includes screening of thousands of the chemical database. Example PubChem. This is the most common height throughout screening. Then rational drug testing. Design and make new chemical according to structure of target physiobiochem. For example, we have discussed omeprazole, proton pump inhibitor by rational drug designing. So when we are discovering the drug, it is not called drug, it is called potential drug or a lead. Then after that we do its animal testing. So preclinical trial, it is also called as non-clinical trial. Animal testing, the purpose of it is safety and only safety, not efficacy. Various toxicities are studied in animals. Single dose toxicity study find acute toxicity. When we give one single dose and we find acute toxicity. When multiple dose toxicity is also found, chronic toxicity or subacute toxicity. What we observe is nephrotoxicity, hepatotoxicity, cardiotoxicity, genotoxicity, cardiogenicity, and sacrifice animal. To observe all these, we sacrifice the animal and observe the organ under microscope. Principal trials, preclinical trials. So the preclinical trials, we follow GLP guidelines. GLP is good lab practice. So there is a rule that at least 2 or more than 2 different species must be tested for toxicity. Because previous to this law, thalidomide disaster has happened. Thalidomide is a teratogenic. It was safe in pregnant rates, but in the humans, when we give for morning sickness that drug thalidomide, it produces phocomelia in the fetus. Phocomelia is short limbs. So there must be at least 2 or more than 2 testing of different species toxicity. These are rodents or non-rodents. So this is important. Rabbit. In rabbit, we take the weight of rabbit as 1.5 to 1.8 grams, and in mice, rat, and guinea pig, these weight are equivalent to human of 60 kg. Preclinical trials is here the drug is known as HIT. Now chemical trials, important topic. The human testing of any drug occurs here. We follow 2 guidelines, GLP and GCP, Good Clinical Practice Guidelines. We follow this ethics, autonomy of patient. The patient, whenever patient want to quit, they quit, no questions asked. Beneficence. We do it for benefit of human, not for disaster of human. No, we don't do it. We do for benefit of human. Then we must take consent, and patient must be of confidentiality. We give them patient numbers and not the names. We should do no harm to the patient. We must think that drug is not better, so we don't falsely we don't do false positive in drug testing. If by any chance the patient is dead, then we must do justice to the patient. We must compensate to his family. So history of ethics, the declaration it was in the Declaration of Helsinki. First ethics. Then ICH-GCP was formed, International Council of Harmonization, good clinical practice. In India, we had enforced a law in 2019, of which is NDCTR, New Drug Clinical Trial Rule. Before 2019, Schedule Y, and now it is NDCTR. According to GCP, clinical trial are also known as phases. So first there is phase 0, which is not mandatory. Phase 1 to 4 are mandatory. First phase 1, phase 2, phase 3. After phase 3, the drug is market approved. It is given to mass population. First phase 4, phase 0, also known as microdosing trial. We give very little dose of the drug, radioactive drug, to identify whether the drug is attached to the receptor or not. The purpose is only target organ binding, pharmacokinetics. We are not studying here the safety or efficacy. So the number of participants is 2 to 5. It is not mandatory. First of all, types of patient needed are healthy humans. Design open label, no binding needed, no blinding needed. If fail, we can avoid phase 1, which is very costly. We give very low dose of radioactive radio labeled drug, and we do by radiation, we do PET scan and mass spectrometry to confirm whether the target organ binding is done or not. Then phase 1, it is called human safety and pharmacology trial. First in human trial, first mandatory human trial. Number of patient needed is 20 to 50. Number of person needed, healthy person. Here healthy persons are required. Healthy person. So here the drug is open label, means no blinding is done. Then safety. Here the purpose is only safety. We identify how much dose is toxic effects are produced. We identify here TD50. We find maximum tolerable dose and find pharmacokinetics, right? So here, for example, after 40 toxic effects is coming, so safety limit is 40 tolerable, maximum tolerable dose. Safety is only purpose in phase 1. Maximum tolerable dose we find out. Phase 1 is not for efficacy. No, it is not at all for efficacy. It is only and only for safety, to find maximum tolerable dose in healthy individual of 20 to 50 numbers. For example, there is an exception. In anti-cancer drugs, we don't give to healthy people. and they are very toxic. So directly phase 1 is done to the patient in anti-cancer drugs. So phase 2 is also known as therapeutic or efficacy exploratory trial, also known as therapeutic or efficacy phase 2. The number is 200 to 500 persons are needed. Here the persons are patient itself. Yes, in this blinding is done. It means patient doesn't know whether he is receiving the drug for his treatment or not. We give some people placebo drugs, no, not placebo, but the drug which is not related to their condition. It is called as blinding, random control blinding. To find the purpose here in phase 2 therapeutic trial is only to find dose range at which drug is producing therapeutic effect. We find a range here and explore the efficacy. So in phase 2, first time we find here efficacy, also known as proof of concept trial. We find here range. It is also known as proof of concept trial. So phase 3 is a confirmatory trial, therapeutic confirmatory, to confirm the efficacy at which the drug is performing well. So number is this much persons are needed, 2000 to 5000, and they are patient. Yes, the blinding is done. Purpose is to confirm efficacy, means actual dose, and also the safety. Phase 3 is multi-site trial, means it is not limited to one place. The drug is transferred to another countries to another patient, because we cannot find 5000 people of same disease in one selective area. So phase 3 trial, after it is done, phase 3 the drug is market approved. So phase 4 is done after market approval. So here after phase 3 drug is market approved. Here now phase 4 post marketing surveillance. Number is unknown. Many drugs are prescribed, so we don't know how many patients are taking. So type is patient taking approved drug. Design open label. Purpose safety. We want to find a drug interaction, compliance, and rare and missed adverse effect. If toxic risk, if drug is toxic, in which risk is more than beneficence, the drug is banned. And if drug is toxic but benefit is more than risk, so toxic effect added label, means drugમાં લેબલ કરી દે કે શું કે બુકમાં ને બધે લેબલ કરી દે કે ભાઈ આ ટોક્સિક ડ્રગ છે પણ આનું બેનિફિટ છે. So ethics can't be applied to phase 4. No consent. There is violation of consent. Okay, as drug is already approved by the government.